Today, it's my great pleasure to introduce Mihaela van der Schaar as our speaker. And it's a great pleasure for me, not only because of Mihaela's work in research for machine learning, in medicine, and so on, but also because I'm a great fan of her personally. So I'm very happy to have her here. Mihaela is the John Humphrey Plummer Professor of Machine Learning, Artificial Intelligence, and Medicine at the University of Cambridge, and also a Turing Fellow at the Alan Turing Institute in London. She works on machine learning with applications in medicine, finance, and education, and she's received a number of honors and awards for her work, including an NSF Career Award early on, and also the Owen Prize on Preventative Medicine. So as I mentioned, her research is a constant source of inspiration for me, but also for many other people, because it spans both very technical topics, advancing our theoretical understanding of machine learning, but also in very exciting applications, for example, in healthcare, which is the topic of today's talk. So I don't want to take up any more of our time here today. I would like to hand over now to the speaker, Mihaela. So please go ahead and welcome.
Thank you. Thank you very, very much for a very kind introduction, Frederick. And actually, I'm delighted to be today with you. And I'm myself a very big fan of Frederick's work in this area. So the pleasure to talk to each other is mutual. And I'm really very happy to talk to you today. And what I'd like to present to you today is some thoughts on why I believe that healthcare and medicine are not only interesting areas to apply machine learning and artificial intelligence, but why, even more than that, machine learning in medicine is especially interesting because of the complicated problems that we need to tackle. So I'm going to argue in this talk that medicine is creating new frontiers for machine learning, helping machine learning grow, and obliging us to think differently than we think in conventional areas in machine learning. And we all know that artificial intelligence and machine learning has accomplished wonders on a variety of well-posed problems where a solution is well-defined and the solutions are verifiable. If we want to teach a robot how to do a particular task, it is relatively clear what needs to be solved and how we can identify that this task has been completed successfully by the robot. Even if it is to take a complex task, such as AlphaFold 2, the current protein folding method developed by Google DeepMind, we can see that the problem has been defined and well understood for decades. Of course, Google DeepMind made a big advance by solving it, but the problem was well understood for a very long time, and we knew how to identify whether we have successful solutions. In healthcare and in medicine, this is a lot more complex because the problems are not well posed, and the notion of a solution is often not well defined, and the solutions are hard to verify. So we need to think out of the box. If we want to identify early whether a patient is going to deteriorate and, for instance, develop dementia, or a patient may develop cancer and we want to identify whether this cancer has been developed early at an early stage in order to treat effectively. These are very complex problems to formalize and think about. And even if we think about the interaction between clinicians and artificial intelligence systems, this also represents an important complex problem. So this represents enormous challenges, but also enormous opportunities, pushing the frontiers of machine learning. So I'm going to argue today that it is really healthcare that drives not only a revolution in formalisms, but also in terms of models and techniques in machine learning. So our group's mission is to develop machine learning and artificial intelligence methods to turn medicine from an art to a science. Machine learning to deliver bespoke medicine, to understand the basis and trajectories of health and disease, to empower healthcare professionals and patients, to inform and improve clinical pathways, better utilize resources and reduce cost, transform population health and public health policies, and enable new discoveries both in the clinical setting as well as new therapeutics. In order to solve these various problems in healthcare, you really need to think out of the box. This is why our lab does research in machine learning at the fundamental level on many domains, from automated machine learning to time series forecasting, from causal effect inference to causality, from reinforcement learning to inverse reinforcement learning, as well as machine learning interpretability and trustworthiness. And often we combine ideas from statistics and machine learning, pushing the envelope. If you want to see the variety of topics one can work on in healthcare, please take a look at the NeuroRIPS 2020 conference, where you can see that our lab has worked on many different types of problems, all in healthcare, but pushing the boundaries in machine learning in a variety of different domains. Today, I'm going to mainly talk about AutoML, causal effect inference, and interpretability. So first, in order to develop clinical analytics at scale, we often face the following problem. We have a particular data set and we need to provide the best machine learning model to build this analytics for a particular problem. So for instance, we want to detect cancer or we want to predict mortality for a particular subpopulation. And this is difficult for a medical researcher or for a clinician because different models may be best for different problems. And they may not have the analytical machine learning expertise to choose the best problem, the best solution. Hence, we need to provide them with the right tools to select the right machine learning models to employ for their analysis. But these models also need to be reproducible, interpretable, and trustworthy. So one way in which we can attack this problem is by using automated machine learning. This is useful because there are many diseases and many variables that may play a role in a particular development of a clinical analytic, and all may be changing over time. For instance, now in the era of COVID-19, we see that the disease is evolving and maybe different populations are affected and the way we treat them is different. So we need to have the ability to build fast analytics to empower clinicians and patients. And we cannot do that again and again and again in a brute force way. This would be both slow and prohibitively expensive, and also at times expertise is not available in some of these medical centers or healthcare authorities. So instead, what I propose here is to use machine learning itself to do the crafting of clinical analytics. Thereby, to some extent, I propose to replace us, the machine learners. As soon as a problem is well defined and well understood, it's nice to have machine learning itself take over our own role and build such analytics instead of us. Of course, automated machine learning is a very rich area and an established area in AI. However, in healthcare, there are different issues and different problems that go beyond what AutoML has solved in other domains outside healthcare. So I'm going to highlight a few of them today. And one of the differences is that we have a huge design space to cover because we deal with a variety of data sets that may require not only predictions, but also need to deal with missing data imputation, as well as feature and dimensionality reduction. So there are many possible pipelines that we may need to build to issue such clinical analytics. We have done a first version of such an automated machine learning framework a few years back. We call that autoprognosis, and this is a tool for crafting clinical scores. What it does, it takes clinical data as input, and then at the push of a button is creating pipelines that are formed of imputation methods, feature processing, classification, and calibration. It selects among the different algorithms that are available at every stage in the pipeline, the best algorithm and hyperparameters in an automated way. And then it creates multiple pipelines that it integrates using an ensemble construction method. That's a Bayesian approach that we are using, thereby issuing predictions. We also issue explanations associated with this. I'm going to talk more about interpretability later. Another thing that we are doing in subsequent versions of autoprognosis is to learn from other data sets, from other locations, either other hospitals or other countries. So these other hospitals or other countries may have complementary cohorts, which may be different. There may be distribution mismatch and feature mismatch between the current clinical data and these complementary cohorts. So we need to learn through transfer learning and meta learning how to incorporate this additional information into the current pipelines. And we do so in a subsequent set of papers that are complementing the autoprognosis. I'm not going to dwell a lot on this, but I'm going to say that for us working on machine learning for healthcare, it is really important to develop technologies like this one, such that we empower clinicians to be able to use these technologies easily off the shelf. What we have done with autoprognosis since its inception is we have applied it to a variety of areas. Initially cardiovascular disease, cystic fibrosis, breast cancer, and most recently together with the National Healthcare Service here in the UK for COVID-19. So it is useful to have, again, such automated machine learning models. But this is not enough. For healthcare, we cannot just stop there. We need to go beyond prediction. We need to be able to do time to event analysis, survival analysis. We need to deal with competing risks because the patients may not have only one disease that they may be at risk of, but maybe multiple underlying diseases. We also need to deal with treatment effects and causal models, as well as longitudinal data. So in a subsequent set of works that I'm not going to highlight today, but you can take a look if you are interested, we have developed an entire suite of AutoML methods for all these different areas. And if you take a look at these different works, you are going to see that each one of them poses new and different challenges, as opposed to, for instance, autoprognosis. So every problem is different and requires a different way to think about automating the crafting of analytics. Today, I'm going to talk only about how we can do AutoML for causal models. You probably are familiar with this type of setting because you know the wonderful work that Frederick has done in this area. I'm going to just briefly outline the problem to remind you about it. So the focus here is on estimating from observational data the effect of a treatment or intervention for an individual. And the reason we want to do that is because we know that treatment effects are often heterogeneous across the population. Let me say that currently, people are not using often observational data to determine this, but rather the golden standard is of developing and performing randomized controlled trials. However, randomized controlled trials have multiple deficiencies. One of them is that they often do not employ representative population. So if you look at the trials for COVID-19 vaccines, you are going to see that the young people and elderly or people with many comorbidities are often not enrolled in such trials. Yet when the vaccine is enrolled, it is given also to such population. But this is not only true for COVID-19, it's true in general. Usually these randomized controlled trials are expensive, so they cannot be run on a vast number of patients. So the disadvantage is that it may be very difficult to identify really what treatments are useful for what subclasses of patients solely from randomized controlled trials. That being said, randomized controlled trials are and will remain the golden standard for identifying effectiveness of different types of interventions. What I want to bring to your attention is the fact that there is a lot of work that machine learning can do to empower clinical trials. So one of the new frontiers is using machine learning to build adaptive clinical trials, identifying which patients we should recruit in trials, how to determine the dosage of the patients in the clinical trial, how to determine subsequent recruitment of patients if you identify that the treatment is more efficacious on a particular subclass of patients. Here you see some of the work we have recently done using machine learning for adaptive clinical trials, but I think that a lot more can and should be done in this area. So I want to bring this to your attention as an interesting area for machine learning to make a difference. And it combines ideas from both reinforcement learning with ideas from causality. So it's an interesting area. And if you want to see the impact of what machine learning can do in clinical trials, please take a look at the recent paper we have written together with a few collaborators from the pharmaceutical industry. And you can see the role of machine learning not only on COVID-19 medication and treatments, but also what machine learning can do if clinical trials, for instance, for cancer are stopped because of COVID-19 and need to be restarted, how to deal with these problems. So you can see here the impact and the potential, hopefully motivating you to think about interesting problems that you can tackle using new machine learning methods. Today, I'm going to talk about the setting that's maybe more familiar to you, learning from observational data, which has the advantage that we have much more data available to it, and we can learn at scale in a fast and cost-effective way. And as Frederick has mentioned, this has been an area of interest for us for quite some time. And if you want to see the entire work we have done in this area, and you want to see the different aspects we have looked at, we have recently developed a website dedicated to our work in this area, since this is one of our main research pillars in our work. And we do work that is both similar, but also some of it is complementary to the work of Frederick. So please take a look and let us know what you think. We will be delighted to engage with you. So as you know, causal effect inference is a complicated machine learning problem, more complicated than supervised learning because we do not observe counterfactuals. We only observe the factuals. So if a patient, for instance, the patient here in red, which has features X, is treated, we only observe what has happened to this patient if he indeed is treated. We do not observe the counterfactuals. So due to this reason, this becomes a complex problem. Another reason is, of course, the fact that because this is observational data, we need to deal and model bias as well. A lot of work has happened in this area over the last years. Most prominently, a lot of work done by Frederick himself, seminal work in this area, seminal pioneering work in machine learning. We have done subsequent work in this area. And here you see in purple, with a purple arrow, some of our own work in this area. And this is only a very small sample, a small bias sample of the work in this area. And what you see is that different methods have been developed using a variety of machine learning methods, from more conventional neural networks to maybe GANs, to Gaussian processes, but also to trees and other types of machine learning methods. The challenge I'd like to address today is we have all these wonderful methods, and groups such as Frederick's and mine continue to push the boundaries in this area. But the challenge for medical researcher or clinician that wants to use these methods is which model to use, because we have so many and so many interesting ones. And again, the challenge that I like to address is how can we select the best model for a particular observational study? How can we use AutoML to help the clinician select the best model for this particular data? And this is the problem that we have tried to address in our ICML 2019 paper. How can we automate this problem? And the reason this is a more challenging problem than for autoprognosis is that here we cannot do easily cross-validation because we do not observe these counterfactuals. So how can we deal with this? Usually we estimate the performance of the causal effect inference models using the mean square error of the causal model. and we compute what is known as the precision in estimating heterogeneous effects, or for short, PEHE. But how can we validate the causal model without observing the causal effects? How can we do that in order to be able to select the best algorithm for a particular data set? The insight we have used in our ICML 2019 paper comes from a very simple idea: Taylor series approximation, which you are all familiar with from high school. And the idea here is that the value of a function at a given input can be predicted using its value at a proximal input. In this way, we can approximate the value at a new point. In an analogy with the Taylor series approximation, we are going to do an approximation of a PEHE, of the precision estimating heterogeneous effect, but this time we are going to compute the performance of a causal inference model, which is no longer a functional, it's no longer a function, but rather a function of a function, is a functional of the data generating distribution. So what we are going to do is we are going to approximate the true distribution using the synthetic distribution with known counterfactuals. We are not going to use the Taylor expansion, but we are going to use an analogous with it based on von Mises expansion. So we are going to use functional calculus for that. And this is nothing else than intuitively a distributional analog of a Taylor expansion. And we are going to replace the derivatives with influence functions. In this way, we are going to be able to predict the performance of a causal inference model using influence functions of its loss on a similar synthetic data set. So how are we going to do that? We are going to synthesize counterfactuals in two steps. First, we are going to do plugin estimation, where we are going to determine the plugin model and compute in the plugin PEHE loss, and then we are going to do the bias correction. In this way, we are going to be able to estimate the loss, which is actually, in this case, the precision estimating heterogeneous effects, which is, for this problem, as I mentioned, an inaccessible empirical measure. You cannot compute it directly, but we can approximate it. For instance, here highlighted with the first order Taylor approximation, but you can go beyond the first order Taylor approximation. But here for illustration, just the first order Taylor approximation by using the accessible empirical measures as computed in the previous slide. In this way, we have access to the synthetic plugin distribution and the influence function associated with this. So we can in this way compute the true distribution or an approximation of it. It is this approximation of the true distribution and of the true PEHE loss that we are going to use now to be able to estimate the performance of a method on this new data set. So what we have done, we have taken this Atlantic competition, which has 77 different types of benchmark data sets. And first, what we saw is that if we run the different methods on them, there is no one method that's best for all the data sets. So less than 20%, even the best of methods, are not at that time that we did this analysis, which is 2019, so almost 2 years ago, there was no one method that won more than 20% of the time. However, this automated machine learning framework based on influence functions was able to select the best method out of the many methods that we incorporated, many methods of ourselves, but also Friedrich's methods, as well as other methods from the literature. And in this way, 72% of the time, the best model was identified. So this is an interesting way to think about models and model selection. This does not, though, explain why a model is best. This selects methods, but doesn't go to the explanation as to why. If you want to see some more recent work from our lab in this area, we have a recent AI Stats 2021 work where we again go in this direction, but we don't do automated machine learning. We try to understand why certain architectures are more efficient and when. And we build this analysis and fundamental understanding on the basis of several methods developed by Friedrich himself. So if you want to select the best method, this automated machine learning framework, I think, does the trick. If you want to understand why certain methods are better and maybe use this as a guidance to develop new methods, I'd like to point you out to our recent AI Stats 2021 paper and more about our work here. Okay, so I already talked about causal models. I'm going now to highlight a recent work we have done for longitudinal models. And I have been for a long time working on longitudinal setting, both for predictions, for treatment effect planning, but also for monitoring. And one of the disadvantages of this area was that every time I would develop a new method, I would need to implement a whole set of other methods that were developed in the literature. So what we have done in recent times, and this work has been accepted to ICLR 2021, is to build a unified end-to-end pipeline for clinical decision support in the longitudinal setting. And this paper not only provides a suite of methods, but also an entire software suite. So if you are interested in looking at longitudinal settings, you can use clairvoyance as the baseline for you, and you can build on top of it. You can do benchmarking, and you can highlight why your method is indeed best. So I think that this is an important step towards having a more integrated approach across the various groups working on machine learning for healthcare, such that we can easily benchmark and improve on top of each other's ideas, thereby moving the area forward. So I think that clairvoyance is an important advance, especially in terms of reproducibility, because now we can have at the push of a button a variety of methods being compared. What I'm going to highlight today is different modules in this pipeline. This framework contains both static and dynamic data imputation, predictions, treatment effects, active sensing, interpretation, uncertainty estimates. But today I'm going to mainly focus on treatment effects over time, active sensing, and interpretation. So we talked already about treatment effects in a static setting, but we can do that also in a longitudinal setting. And this is what we started to do a few years back. So in this setting, unlike in the previous one, I don't have only baseline information. I also have longitudinal information that's acquired over time. And I'd like to be able to estimate and issue decisions of what's the best treatment plan to be used for this particular patient, given his or her patient history so far. So for instance, here what you see is an example from the area of cancer where I may have multiple types of ways to treat a cancer, for instance, chemotherapy and radiotherapy. And I would like to decide for this patient, given the information so far, whether I should use chemotherapy or radiotherapy, but also when I should start treatment and when should I stop treatment. So I would like to look over time in the future, what's the best treatment to use, which includes the timing of starting the treatment or stopping the treatment. This can be done for one treatment or multiple treatments simultaneously. So unlike in the previous setting, I'm having now a longitudinal patient observational data that contains patient-dependent patient features, as well as time-dependent treatments, as well as the static feature variables like I had before. This represents the patient history. I also have outcomes. Of course, like in the previous case, factual outcomes. I do not observe counterfactuals. So on the basis of the history of the patient so far, I would like to make predictions in the long run, in the short run, in the long run, as to what will happen to this patient if I use different type of treatments. And as time progresses, I'm observing the results of the current treatment, but not counterfactuals. Yet I'd like to learn what will happen and what would have happened given different types of treatment plans. So we set this problem still in the potential outcome framework, but especially now anchored in the wonderful work of Robbins and Hernan. And they have modeled this potential outcome in the dynamic setting. And again, we need to make here two assumptions that we make them also in the static setting, but here they are more involved, that of sequential overlap and strong ignorability. In a 2020 paper, we started to look at relaxing a little bit under certain assumptions these constraints. So we have a paper called Time Series Deconfounder by my student Ioana Bica. So if you are interested to learn more about the frontiers on confounding and deconfounding and hidden confounders, please take a look at Ioana's 2020 paper. I will not talk about that today. Today, I'm going to make these two assumptions. And the challenge is, even if we are making these two assumptions in these dynamic longitudinal settings, are that the patient history contains time-dependent confounders, which are going to bias the treatment assignment in the observational data sets. And these patient covariates are affected by past treatments, which then influence future treatment outcomes. So we really have a complicated situation here, and we need to learn in the presence of this bias from the time-dependent confounders. One way in which Robbins and Hernan have formalized this problem and started to think about this problem is by applying inverse probability of treatment weighting. And this inverse probability of treatment weighting is going to create a pseudo population where the treatment probability no longer depends on the patient covariates. And in this chosen population, the counterfactual estimates are unbiased. In the original work, the marginal structural models, this was done using statistical models, and in a paper much later, a few years back, I and a few of my students have kind of built on top of the marginal structural models using recurrent neural networks to improve the performance of marginal structural models and enable them to deal with a lot more complex variables and learn much more powerful representations. However, the disadvantage of both the original marginal structural model as well as our work that built on it is the fact that this inverse propensity treatment weighting are suffering from numerical instability when the treatment probability is small, as well as high variance due to the extreme weights. So a solution that we have proposed last year was to improve on top of this by learning better representations and balance these representations to create treatment invariant representation in the time series setting using counterfactual recurrent neural networks. So what do we do here? We are building treatment invariant representations using a domain adversarial training, and I'm going to go a little bit more into that in the next slide. And we are building for that on the work on Ganin. And what we do here, we are estimating the counterfactual trajectories using a sequence-to-sequence architecture as shown in the bottom of the page here. And the sequence-to-sequence model, the encoder is using domain adversarial training to build at each time a representation of the patient history, but that is treatment invariant. And this breaks the association between the patient history and the treatment assignment, thereby removing bias from the time-dependent confounders. And then this representation is passed to the decoder, which is continuously updating while estimating the outcomes. Again, we are training this using adversarial balance representations by extending the framework of Ganin. So Ganin proposed an adversarial framework for domain adaptation, which involves building representations which achieves a maximum error on a domain classifier and a minimum error on the outcome predictor. In this framework that we have built for counterfactual recurrent neural networks, We use domain adversarial training to build a representation of the patient history that is both invariant to the treatment given at the time step t and that achieves low error in the predictive outcome, in the treatment outcome. We have used a semi-synthetic data set to evaluate the performance of such a model. And for that, we are using a Nature paper that was published a few years back. And this is interesting work because it's very parameterizable and allows us to do interesting synthetic experiments because the parameters of this model are allowing us to control in a very clear way the time-dependent confounding. I will not go into the details of the performance, but let me just say that this counterfactual recurrent neural networks do better than both our own method based on recurrent neural networks, as well as the marginal structural models, the initial methods. And this is true in both treatment accuracy as well as treatment timing accuracy. If you want to play with this framework and build on top of it and improve it, please take a look, or at least benchmark against it. Please look at our clairvoyance framework. Now, let me go one step further and talk also briefly about active sensing. Why is active sensing important? Because we want patients that may have been diagnosed with a particular disease or patients that are at risk of a particular disease to be screened effectively. So we want to move from a one-size-fits-all screening, where, for instance, a patient with cancer is asked to come back every 6 months or every 3 months, to a setting where we identify which patients need to be screened more often and with what technology and which one maybe less often, thereby doing this in a much more efficient way and without the side effects associated at times with this very intrusive screening. For that, we developed a suite of methods that you can find in clairvoyance, inclusively deep sensing. So deep sensing is able to estimate what's the value of information for a particular patient at a particular moment in time. So the reason this work is interesting, and I want to bring to your attention in hope that maybe you can come up with even better methods, because I think that this is an underappreciated area, the area of personalized monitoring and screening. So here what we do is we are doing this active sensing using a multi-directional recurrent neural network. So what are we in effect doing is we are learning the value of information for a patient over time. And this is challenging because what information we should acquire, when, and what is its value for a particular prediction or treatment effect estimation is unknown and is changing over time. It needs to be learned. So what we need to do ideally is to learn at the training time the value of information. And we may want to learn it using a neural network that learns at various cost performance points. And we can do that by removing data at training time and learning representations at different cost performance points for different patients. These representations are learned and constructed then recursively and adaptively by deliberately introducing missing data, which enables us to have these multiple cost performance points. Hence, at training time, we learn representations that are based on different levels of missingness. And then at runtime, we are learning when to acquire the information next, because we have this wealth of representations that are already available to us, and we can use this information to predict whether acquiring a certain particular value at the next time point or in the next 10 times points will be valuable, and how much would this add to the predictive accuracy. So we combine in this way ideas from missing data and value of information with ideas from predictions and counterfactual estimation. I believe a lot more can be done in this area. So this is why I present this work to you today. And if you want to benchmark against it, you can look at clairvoyance. Finally, I'd like to talk just very briefly about interpretations. So we cannot have only predictions in healthcare. We also need to have interpretations, trustworthiness, and explainability. So, for instance, if a particular patient is predicted by, for instance, autoprognosis to have COVID-19 mortality that's very high, we would like to understand why this prediction has been made by autoprognosis for this patient. So we want to X-ray the black box model. One way to do that is to use feature importance, personalized feature importance, to identify what features were important for the black box model to issue this prediction. And it may be that for Mary there are 2 features, but for Bob there are 5 features and different features than for Mary. We have produced for that work that we call InVase. And again, I'm bringing this work to your attention because I think a lot more can be done using these ideas. The reason InVase is interesting is because it does feature importance at the individualized level, not in the conventional way, but rather using reinforcement learning, using a simple way of reinforcement learning, which can be, I think, improved. And that is based on actor critic. So what does it do? It takes a black box model, and then the actor is selecting as actions the number of features it thinks may be relevant for this particular patient. So the input is the features, but the policy, the actor is selecting as actions the features that it thinks relevant for this black box model to look at for this patient. Then the critic is computing the reward, which is the loss between issuing predictions on the basis of all the features versus only the features of this particular model. And this error, this loss, this reward is then fed back to the actor to teach the actor how to select better features in the future, thereby learning what features are relevant for this individual prediction. InVase is similar to Lyman learning to explain in the fact that it is doing individualized feature processing, feature importance in a model-independent way. But unlike these models, it has 2 advantages. One of them is that it's learning the set of relevant features. So it's not only learning what features are relevant, but also the number of features that are relevant. A second advantage is the methodology is very intuitive. Through reinforcement learning, we are learning what features are important. And I think that that's a good idea that can be used in other settings as well. This is why I brought it to your attention. However, for medicine, this may not be enough. Just identifying what features are important is not enough. What we have learned when trying to implement these methods using autoprognosis was that just feature important was not enough to achieve approval for practice in clinical settings. What we also need to provide is transparent risk equations associated with the black box models. These are mandated, for instance, by the American Cancer Association and also a variety of clinical practice guidelines in the United Kingdom. So the question is, can we do feature importance, but more than that, can we understand statistical feature interactions? Can we do a transparent risk equations? And the answer to that is yes. I'm not going to go into the details in view of time, but I'm going to just tell you that in a recent NeoRIPS paper, we are able to use symbolic metamodels to identify white box models associated with black box models. And these meta models are models of our models that transfer, if you like, a black box model into a transparent white box model that is selected, for which the model class is selected by the user. So for instance, the user may decide that polynomials is what they want to see, and the symbolic meta model is able to do so. Is able to take a black box model and provide for it a white box equation. I'm not going to go into the details of this because I was a little bit slow and I will not have time to go into the details. But if you are interested to see more about this work, please take a look at one particular talk that I gave last year, just at the beginning of the pandemic, where I talked to a variety of clinicians presenting a variety of machine learning methods, inclusively the symbolic metamodel. This is a Turing talk that you can find online. And this is one of the 9 papers that we have at NeoRIPS. So finally, I just want to say that, again, predictions may not be enough. We also need to have confidence estimates associated with these predictions. And for that, the usual way to do uncertainty estimates in the machine learning literature, such as Monte Carlo dropout, is not enough. And the reason it's not enough is because we need for machine learning post hoc methods. So we want to understand how confident is the machine learning method in these particular predictions. So we want to compute trustworthiness in a post hoc way. And even more importantly, we don't want to have only coverage estimates, but also coverage guarantees. So we need frequentist coverage guarantees. So in a suite of recent papers, both for the static setting as well as for the dynamic setting, we developed methods that provide frequentist coverage guarantees for predictions. Again, this is not work that it is completely done. A lot more needs to happen. And again, an area that I would like to bring to your attention because many new methods are needed. Finally, if you are interested in what our group has done on COVID-19, since we are a machine learning group for healthcare, please take a look at our research website, where we have early on in the pandemic identified these 5 areas where machine learning can help with COVID-19, ranging from clinical trials to managing limiting resources to managing uncertainty. So you can see the original paper where I have outlines what areas of in which areas machine learning can make an impact, and you can see what we have done since then. Finally, I don't think you need convincing, since you all know the interesting work of Frederick, but I'm going to still remind you that this is a very interesting area, not only because it enables us to think out of the box and develop new machine learning methods, but also because it can catalyze a revolution in healthcare. If you want to see more about the research in my group, but also the research we do in Cambridge, I'd invite you to take a look at the work that we have started to do in the recently formed Cambridge Center for AI in Medicine. We just have had an inaugural event less than a month ago, so please take a look and see the research we are doing that is trying to break frontiers at the interface between machine learning, medicine, and bioscience. Finally, we are hiring brilliant students and postdocs. So if you are interested in this area, please reach out. And also, we are trying to build a community of machine learning students and young researchers, especially now in the pandemic, because my students cannot go to conferences and cannot meet you. So if you are interested in machine learning for healthcare, we started a session that we called Inspiration Exchange, where we present work from our lab and we engage in discussions with you. So if you are interested in this, please go to our website and subscribe to this event series. Thank you very much.
Thank you so much, Mihaela,
A little bit long, sorry, in my enthusiasm, maybe too much.
Oops. I think everyone was very happy that you went a little bit over, so we could see even more of your work. So we have a question about that, actually. There was no coincidence that you struggled for time because you had a lot to talk about. And we have a question from Jens. He says, your lab is incredibly prolific. Can you share some key success factors? What do you do differently from other labs? Recruitment, supervision, choice of problems?
Thank you. Thank you for this question. So first, I need to say that our success is due to the fact that I have brilliant PhD students. And let me tell you something about that. We are very fortunate in Cambridge because we are in our group allowed to recruit students across a variety of disciplines. So the students in my lab come from very diverse backgrounds. I have students who have studied computer science, engineering, pure math, applied math, physics, and economics. So one advantage of my current group as opposed to UCLA, where I was a professor before, where I needed to recruit mainly from the area of engineering because I was an engineering professor. One advantage currently is that our group is very diverse. And I think that in machine learning, the fact that you have students with diverse backgrounds coming at this with different areas, and I think I even forgot to say statistics. We have students, of course, coming from statistics. So the fact that we come at this from different areas enables us to think out of the box and push boundaries. So this is, I think, one important reason. Secondly, we are often interested in solving real problems in healthcare. And as Frederick will tell you as well, it is often complicated to take a tool that exists in machine learning and just beat a problem in healthcare. If you do that, you are going to achieve some success, but not tremendous success. So the more you understand the problem and the way to think about it, the more you realize you need to think out of the box. You need to formalize maybe a new problem with new assumptions, and then you may need to develop new machine learning methods. So for instance, we have developed many different types of generated adversarial networks, many different types of GANs. And the reason these GANs were very different is because we needed to do data imputation with them. We needed to do transfer learning with them. We needed to do synthetic data generation. All problems in healthcare, but which required innovations on GANs. And the same is true in other areas. So all I'd say is recruit diversely, and medicine is a great area to push the boundaries in machine learning.
Thank you. So we've got a follow-up question about that, actually. Do you recruit also from medicine or surgery, for example? There's a question by Hassan here.
Thank you. So we do not, in our group, our students come from a quantitative background. However, we are collaborating with clinicians and medical researchers. So for instance, in this Cambridge Center for AI and Medicine, we have collaborators that are clinicians and MD PhDs, and their groups have PhD students and medical students. So we have collaborators from the medical area, but not in our group. So this is how we interact.
I see. Thank you. So I'm going to jump into a couple more specific questions now about the slides that you presented in your talk. And there were a couple of them about AutoML specifically. I think this was back on slide 23 or something like that in connection with AutoML. The question was, how long does it take to do the variable selection search? Or the selection search was the question, but I'm assuming it's variables. Is the AutoML model much more expensive computationally? It takes more time, or is it something you can do in parallel? Can you say a little bit about that?
So this is actually a very good question, and I went very fast through it. And if you want to hear more about that, we have a variety of talks on AutoML. So if you look at the inspiration exchange engagement session, please take a look at some of these earlier sessions in September, October, and you'll see a lot of discussion about AutoML, inclusively the question you asked. But let me give you a brief answer. So the challenge here is not only the variable selection, it's the fact that we need to do missing data imputation, variable selection, and prediction or survival analysis. And these different modules, if you like, these different stages of the pipelines interact. So the problem becomes combinatorial because depending on the variable selection, what follows afterwards, the machine learning model may be completely different. So the exponential increase that we needed to deal with in autoprognosis was due to the fact that these different methods interact. So it's not only variable selection, it's the fact that depending on missing data before and what follows after, the performance of the feature selection method changed. So the way in which we deal with that, so we needed to deal with that because conventional Bayesian optimization will not be able to deal with more than 20 something dimensions. And we have here hundreds and hundreds. So what we do is we use an innovation. We learn a structure kernel on top of this that learns which algorithms should be jointly optimized and which ones should not. So we learn it to some extent which algorithms need to share learning and learn jointly. And hence, when I get an observation, because how do I learn in AutoML? I try the method with that parameters and I observe the performance. And then I need, on the basis of that just one trial, to update everything else I know about the performance of all the other methods for this data set. Doing that effectively and grouping algorithms that can share information, and hence I can jointly update their performance, and algorithms that do not, is what we do to this using this structured kernel learning. Please take a look for the details in the paper of ICML 2018, Ahmed's paper, or take a look at our various talks on autoprognosis online.
Thank you so much. So we have a couple of questions from Morteza now. Would you like to ask them yourself? I should be able to find you and let you unmute. Or at least start with one, perhaps. Is he still with us? Yes.
Hi. Thanks a lot for the very nice presentation. I enjoyed it a lot. So I'm colleague of Fredrik. I'm mainly working in sequential decision making, so my questions are about this kind of aspects. My first question was about this individualized treatment. When you have the x-axis, which is the time, I was just wondering if this is like the actual time or it's like just order information, like the order parameter. And is it important to distinguish these two or not?
Very, very, very good question. So first, in this time series setting, where we have treatment effects or even just predictions, often you are correct that we may have moments in time when we have observations and moments in time where we don't have observation. The data may be missing. So we are learning in the presence of missing data. So we may have that at a particular moment in time, for instance, at month number 1, we have an observation, and then for a couple of months, we have no observations, then we have again an observation. So we are able to learn in the presence of such missing data. How do we deal with that? We either do time series data imputation, or in the more advanced paper of ours, we learn from the presence of missing data, because this data may be missing not at random, may be informatively sampled and informatively missing. So if you look in the clairvoyance package and what we have done in the counterfactual recurrent neural networks, we do imputation. So if we have missing data, we impute. But in a paper of a few years back, we did missing data not at random in the time series setting, and we use a Hawk's point process to learn from the sampling process. Is that answer clear to you? I'm not sure. So yes, we are learning in the presence of missing data. And if you are smart, you can do something with that missing data. We have done a little on missing data not at random. That's an ICML 2017-18 paper by Ahmed and me using Hawk's point processes. But I think more can be done there.
That's very clear,
It doesn't work. We just did imputation, and if you have a missing value, we just impute. But I am aware that this is not ideal.
Yes, I see. Thanks a lot. That's a very good explanation. So, Fredrik, can I ask the second question, or you want to?
Go ahead. Go ahead.
So my second question is about this active sensing. That is a very interesting topic for me. And is it can we use it also for causal inference?
I think so. So part of the reason I wanted to talk to you all about that is I feel that this is an undervalued topic. So we have fantastic work from Fredrik and David and many others on the causal effect inference. But what we do not have enough is monitoring. And how can we, now that we are using these models to issue predictions over time, how can we make predictions but also decide when to acquire the next sample?
Yes.
We have not done that, but I think we should be doing that. I think it's very interesting. We have mainly done that in our work that I highlighted here, deep sensing and ASAC, for the prediction setting. But I think that this needs to be done. And I think that it's a very interesting problem because people worry a lot about imputation for the data that was already acquired. But I think an even more interesting problem is, okay, this person is sick. We are treating them in a particular way. When should they come back to see if I should continue with this treatment or switch to another? So this is a good frontier. And this can be done dumbly in our current clairvoyance. So we do it in a not very principled way, but I think a lot more can be done and should be done. And I hope I excite some of you to work in that area.
Yeah, that's very interesting and exciting for me too, because I like a lot active learning, and perhaps even it can be combined.
But this is active, not active learning. I just want to say that
active data collection.
It's a little bit different because in active learning, the focus is the labels are expensive. Here is the input is expensive, collecting features. Yeah, so a little bit of a difference there.
I agree. Yes, I agree.
But active learning also interesting.
Yeah, and maybe also it can be combined with active data generation that we were talking about when we don't have enough data and so on.
Definitely, definitely. Actually, that's another, since Frederick, I hope to excite more students at your side about that. Synthetic data generation is another area of big interest in my lab. And part of the reason is we don't have enough data sets that we can use as a community. So often when we do experiments on the same good old data sets such as Mimic or a few others. And even if I have access to some data sets that are private to me, I cannot share them with you to agreements. So I think synthetic data generation is an important frontier. And if you are interested in that, please join us in this inspiration exchange sessions that we are organizing, because this month we are working a lot to engage the community in machine learning for healthcare on synthetic data generation. So generative models for synthetic data in healthcare. Sorry, Frederick, to
No, thank you so much. This is actually answering a different
community to get involved, because until this is again a frontier, I don't think we have synthetic data generation methods that are fantastic. I've worked a lot on it. Other people are working on it, but still a lot more can and should be done. So I hope I excite you all in Chalmers to work on this.
I think so. This actually touched upon a question from the chat that we haven't got to yet, which is about how you surmount the challenges pertaining to healthcare data. And so, I mean, from my perspective, we have a couple of options, right? We have these open data sets that you talked about, or mostly open data sets like Mimic, which are not many. We have synthetic data sets, and then we have data set where we work perhaps intimately with a hospital or an insurance provider or something like that, and we are not allowed to share that data. So yeah, can you say something more about how you think about that? You're saying that you like the idea of synthetic data, but
really
it also
like
has its
the idea of synthetic data. So not many people are going to share data with us, the machine learning community, at scale. So we are fortunate to have adenine, Alzheimer's, Mimic, and a few others, but it's not a lot. And the reality is we need many data sets. We need an image net of data sets in healthcare. And this will not happen unless, because these are sensitive data. This is not cats and dogs. These are very private, sensitive data sets. So I believe that in order for us as a community, of machine learning community, to go further, synthetic data is a good solution. Please take a look at the blog I have written about that. So I've written a blog on my website under Big Ideas on synthetic data. You can see there not only some of our work, but my thinking about the problem. And please engage with us. The reason I believe synthetic data generation with privacy guarantees is interesting, if we can generate synthetic data with high fidelity and high privacy, then we can use these synthetic data sets to run competitions, to benchmark and compare our methods. And then we can take these methods that end up being good and transfer them in hospitals on real data and still make sure that the performance is the best.
Yeah. Thank you so much for that answer, and also for all the references so that we can look up more things after.
Please take a look, and please join us and my students in this engagement, because this is one of the topics we will discuss.
All right. So we have another question from the chat here. I know we're running a little bit over. For everyone who is currently in the session, please stay as long as we keep going. And Mihaela has already offered very kindly to stick around for a little bit longer.
No, I'm very excited. I'm very happy to discuss and engage with you, and hopefully in the future collaborate.
I'll be glad to hear it. I'm going to ask you now a question from Özgür, who asks, how do you use the results from the white box models? So I'm assuming,
Oh, very good.
the goal is to replace something more complicated with something more simple. But how do you
Very
actually end
good.
up using those?
I apologize for jumping very fast through it. I talked a little bit longer than I hoped for. If you want to hear this in detail, again, I invite you to take a look at this Turing talk. This Turing talk is interesting, not only because I go in the details of these white box models, but at the very end of the talk, because I gave this talk at the Royal College of Clinicians, you will hear a lot of discussions between me and clinicians about the topic. So that's, I think, interesting. I will answer your question, but I want to point you out to the talk, not only because I have more time to go into the details, but the discussion at the end is interesting to hear the clinicians think differently than us. Because you know what we find interpretable, they may not find interpretable. But to your question, so we take a black box model and we then create, using the symbolic metamodels, a white box model, which may be a polynomial or maybe a set of Bessel functions, or for different communities, we may have different white box models. Then what do I do is I use that white box model to issue predictions. I take the black box model and I throw it away and I never use it again. So in the end, when I issue predictions at test time, I'm going to issue them not on the basis of the black box model, but rather this white box model. And hence I can interpret the predictions, I can look at the equations, I not only can look at feature importance, but I can tell you exactly how, if I increase a little bit this parameter or this feature, how will the prediction change? So I can really go through this to understand as a clinician or medical researcher, or to explain to the patient. So you may ask yourself at this stage, why do I need a black box model if I throw it away? I need it because it guides me to the white box model. So this black box model is the one that I'm approximating using the white box model. If I would not have had the black box model, I would not have learned how to approximate just from data. So the black box model is not needed at testing time, but it is needed at training time to help me learn the white box model. And I cannot do it just on the basis of data. At least I don't know how to do it.
Thank you. So I wanted to ask a question myself as well, and it has to do with your work on data from the COVID pandemic. And so a lot of people have been engaged in this huge task, like of trying to make sense of how to treat patients with COVID and make prognoses and so on. What are the main takeaways from your work with this? What have you learned about how we should deal with these kinds of situations for in the future, and specifically for this one?
It's a good question, since it's more of a synthesis question. So first, I think one thing that I've learned early on in the pandemic is that we need to try to think out of the box as a community of machine learners as to where we can help. And that's the reason I identified early on many areas, because there are issues that may be less interesting, which may be more about infrastructure, if you like. Can we predict whether hospitals are going to run out of beds? This may sound not very interesting from a problem point of view, but that may be very useful for clinicians and for operationalizing some of these technologies. Also, while in normal times, maybe machine learning may not impact clinical trials, because we need to run these trials at speed, machine learning has a chance to really break into clinical trials and adaptive clinical trials. So I'd say I've learned that we need to think broadly about how we can help. We can engage with a variety of medical professionals and bodies to think where technology can help. This may help us to identify new problems. And also to become humble, because when we started this, we thought we are going to be able to do a lot. But then what we realize is that to operationalize some of these methods at scale require way more than academics groups. We really need almost like an implementation arm associated with it. And that is not often available in academia. So we need to find partners, whether it's startup or industrial partners, to help us scale. So I think that this is something one realizes. So we try to do a lot of implementation ourselves early on, but we realize that beyond the point, we are not able to go beyond that. Another thing is again related to synthetic data. It would have been wonderful to collaborate with other machine learning researchers and share data with them. And this was very difficult to do because we couldn't share the data we had. If we were having well-standardized synthetic data, we could have shared that and maybe would have done more discoveries faster. So again, I think it shows the importance to be able to easily, when a new disease or a new pandemic comes, how can we collaborate fast with each other such that we move at pace and enable others with our technologies. So
that's a very good point. I can mention that we are currently at Chalmers. We are reinforcing, I should say, our collaborations with the local hospital here, Saarbrücken University, and are in the process of streamlining the infrastructure challenges that you're talking about sharing data and so on. And I think this gives us some advice about how to proceed there. But do you have any other kind of
yes,
guidance
actually,
that you would wish to give us?
One other guidance, so I'd like you to take a brief look, if you can, at, I've written a paper with Zhao Zhiqian and Mihaela. That's a machine learning journal paper that appeared in 2020. And there we are talking about the lessons we have learned while working with NHS, the National Healthcare Service here in the UK. And we talk about the excitement, but also the lessons learned, and especially the deployment lessons learned. So I invite you to take a look at that paper. It may not be rocket science from a machine learning point of view. As a matter of fact, we use autoprognosis to predict how many beds are going to be needed in hospital. So in a machine learning point of view, it's not rocket science. But what is interesting there is how we took a technology we had and very fast, in just 1 month, we created an entire simulator and ecosystem such that this technology can be tested in many locations, and the lessons learned, both positive and also the challenges. So if you are interested in more the implementation side of machine learning in hospitals and multiple hospitals throughout the country, I'd advise you to take a look at that paper.
Perfect, thank you. So I'll ask you one last question before we wrap up, and it's also about the synthesized data that we talked about before. One of the questions here is, how do you validate the results that you learn, or the takeaways that you get from working with the synthetic data when you move back ultimately to real data? Have you done any work on that, of trying to transfer models from one together?
Thank you. Yes, this is a fantastic question. We have done a lot of work, and let me tell you about it and hopefully get you excited about that, because that's my goal in this talk today. First, we ran at NeoRIPS 2020 a competition for assessing the quality of synthetic data. This NeoRIPS 2020 competition was called Hide and Seek. Hide because we create synthetic data. Seek because the seekers are going to try to identify patients in this data. Take a look, please, at this NeoRIPS competition and how we design it. But this is not enough. You will see that this competition was very challenging to run, and there were many problems with assessing the quality of synthetic data. In our next engagement session, Inspiration Exchange, we are going to have an entire session dedicated to this topic, where I and a few of my students will discuss exactly that. We are going to talk about what we have learned from the competition, how we have improved our assessment of such synthetic data, and where we think the next steps should be. So again, please subscribe to this Inspiration Exchange and come and join us if you are interested in this, because we need to build a community to think about not only generating synthetic data, but like you rightly say, assessing the quality of it. And we do that in many ways. We do that at times by running competitions on the synthetic data and showing that the ranking stays the same on the real data, but that's not enough. You want to hear more, come join us and discuss with us and debate with us, because we need to figure it out. And I'm not saying that we have figured it out. We have a lot of ideas. We have done a lot of work thinking about it. I'd love to present it to you together with my students and hear your thoughts and get involved.
Perfect. Thank you so much, Mihaela. I think that will do it for the Q&A session.
And I hope to come in person sometime soon.
I hope so too. Hopefully before not too long. So that would be amazing to host you here. And again, thank you for everyone, to everyone in the chat who has asked questions. We got really good ones today, I thought.
Thank you.
We will post. We will post the video of this on YouTube, but also on our websites, where hopefully we can get the slides from you, maybe Mihaela as well.
Sure, Frederick. I can send the slides.
Perfect. Thank you so much. Okay, that does it for this talk. And look out for more announcements from the Chalmers AI Talk series. And we'll see you all very soon. One last thank you, Mihaela.
Frederick, so much. Thank you very, very much.
Bye for now.